Okay, so good afternoon or good morning if you're joining from elsewhere around the globe. Very warm welcome to this session this afternoon, what Nucleus is holding in collaboration with Infinity Biologics. So I am delighted to be joined by two expert speakers on our session this afternoon. So firstly, we have Dr. Mike Sheldon, who is Senior Director Scientific Affairs at Infinity Biologics. And then we also have, or if you want to give a wave, Mike, please feel free. And we also have Ben Roylance, who's VP and General Manager of Strategic Commercial Operations at Infinity Biologics. So thank you, Ben. So during this session, we're going to have an overview of how IBX are going to help you to leverage the potential of biobanks. So really looking forward to the session this afternoon. Just to recap on the format, so it's a presentation format and there'll be time for Q&A. We are a small group, so actually the question box is enabled, but if you would prefer just to unmute to ask your question towards the end of the session, then that's equally acceptable and very happy for you to do that. As I said, we're a small group, so no need to be shy. We are recording, just to clarify that as well. So we're scheduled to run with the presentation and Q&A till about 3 o'clock. But just to stress that our speakers are available to stay on the call for up to an hour afterwards. If there's any elements of the session that you would like to discuss in further detail, so I can actually arrange for some breakout rooms on the session, and I can allocate people into those. I think we could do one-on-one sessions, 15 minutes per person. So depending on your own schedule, of course. If that's not possible and you have to jump off at 3, then I'm very happy to act as a linker between IBX and any attendee that would like to maybe schedule a follow-up conversation. So with that, I'm going to mute myself and go into the background and hand the stage to Mike and Ben. So Mike, I think you're presenting first, so over to you. Thank you very much.
Okay, I'll be sharing my screen with you then. Please let me know if this works well. I was a little nervous with Zoom.
Looks great.
Can you see my slide? Terrific.
Indeed.
Okay. Thank you, Alicia. And good morning, everyone, or good afternoon, depending on where you are today. As Alicia said, my name is Mike Sheldon. Mike is fine. And I'm the Senior Director of Scientific Affairs at Infinity Biologics. Our labs are located in sometimes snowy Piscataway, a town in central New Jersey, about 1 hour away from New York City. My only regret really in not being in today is not being with you in person, and I hope that won't be the case next time. Today, I'm going to offer you an overview of our company, as well as offer an example of how we met a challenge that we've all had to meet as a society, and that is the advent of the COVID-19 pandemic. This story demonstrates the breadth of services and capabilities that we offer to clients around the world. First, a bit about our history. Some of you in the audience today, I see Matthew has joined us from Genetic Alliance, may be more familiar with us as RUCDR Infinite Biologics. Jay Tishfield formed RUCDR in 1996 as a biobank and sample processing facility initially serving the NIH and academic communities. We were at the outset, and still are, the biobank for a number of agencies of the NIH, such as the National Institute of Mental Health, NIDA, and others, as well as large organizations like the Simons Foundation for Autism Research and the Immune Tolerance Network, to name but 2. Years later, in 2012, we formed a strategic alliance with a company that was known at the time as Biostorage Technology and later became Brooks Automation. This strategic alliance allowed us to expand relationships with the commercial sector, working on large-scale genomics and other types of projects with pharmaceutical and biotech companies. The COVID pandemic changed all our lives in 2020. It was very early on in this event that Andy Brooks, at the time COO of RUCDR, recognized the important contribution that large biobanks could and indeed should make, given the infrastructure and intellectual resources available to us. We immediately began work on development of a COVID assay that would be accurate, sensitive, easy to administer, and economical, so that it could be disseminated among as many people as possible, as rapidly as possible. While assays based on the utilization of nasopharyngeal swabs were available, we reasoned that a kit based on saliva collection would be beneficial, both in terms of ease of use, indeed the subject could perform the collection on themselves, as well as in limiting the exposure of healthcare workers to potentially infected individuals. In April of that year, the FDA issued an emergency use authorization for the IBX at-home saliva collection COVID-19 test, the first of its kind. This achievement was hailed by the White House Coronavirus Task Force and New Jersey Governor Murphy as a major milestone in the fight against the virus. 2020 was also a historic year for IBX itself. In August, IBX formally acquired RUCDR Infinite Biologics, fulfilling Andy's vision, that's Andy shown on the slide, of becoming a commercial company with all the increased potential for supporting our clients that entails. This talk indeed is dedicated as a tribute to Andy, who sadly passed away earlier this year, and we're all dedicated to carrying on his vision for the company. In one short year, this effort to establish this company has borne a number of fruits. In 2021, pardon me, we were awarded a contract by the CDC to perform surveillance sequencing of COVID-19 to monitor variants in the population. Launched a collaboration with Ginkgo Bioworks to scale the COVID-19 response with sample pooling. You can see my cursor there. And developed 2 genetic tests for breast cancer polygenic risk score in collaboration with Genetic Technologies, and Alzheimer's cognitive decline risk assessment in collaboration with Cytox. This portion of the presentation will review how IBX leveraged the capabilities of one of the world's leading biobanks to build a facility that has come to play a key role in the response to the pandemic, with high throughput sample processing, testing, and sequencing, as well as vaccine storage and distribution for the states of New Jersey and Minnesota. The story of this development is that of a journey that began in January of 2020 and continues to this day, represented by the timeline on this slide. Several milestones, pardon me, are notable along the way. The beginning was on January 30, when the WHO declared a global health public emergency and the first US case was reported. From that point to April 2020, we worked on developing the saliva test that I told you about earlier. In August of that year, RUCDR Infinite Biologics became Infinity Biologics. And in October, the IBX launched the IBX Minnesota laboratory, increasing our capabilities of supporting COVID-19 testing for the Midwest. All of the testing, SOPs, training, and compliance issues are identical between the New Jersey and Minnesota labs. In February of 2021, we received one of the 6 center contracts issued by CDC for surveillance sequencing of COVID-19. So what were the major components that went into establishing this capability on this timeline? It all starts with technology development, the recognition of a need, in this case for rapid and economical testing, and the developmental resources and experience to bring it to market. We brought together a number of technologies from different sources, to this end. The saliva kit was chosen from Spectrum Technologies, and this was chosen because of its economic ease of availability, and also the preservative solution within inactivates the virus, making sample handling safe in the laboratory setting. Collected samples in these vessels are decapped in our laboratory and transferred to extraction plates using Janus liquid handlers, followed by extraction using Chemagic 360 instruments, in conjunction with the Chemagic viral DNA RNA kit, shown in green here. This choice was critical, as this kit provides reliable extraction of COVID RNA with reproducible and quantitative detection. The extracted RNA is then subjected to quantitative PCR using the Thermo Fisher TaqPath SARS-CoV-2 assay. The validation assays performed at IBX that led to the development of this workflow and subsequent FDA approval are now part of the SOP of the EUA and in use in our laboratories. A technology, however, is only as useful as the infrastructure and people needed to implement it at scale. Logistics is therefore a vital component of the effort. Within approximately 8 months, we were able to secure sufficient lab space, personnel, and equipment to support as many as 100,000 tests per day. The resources needed for this capacity, as you might imagine, are considerable. As many as 300 staff split into dedicated teams responsible for sample receipt and accessioning into our LIMS system, sample aliquoting and preparation using high throughput liquid handling instruments, nucleic acid extraction, quantitative PCR with analysis, interpretation, and reporting of results. And I should add at this point an illustration. The reporting of results was facilitated by the development of Destiny software by our very talented IBX bioinformatics team, and is an important component of the entire flow. We set ourselves a goal at the beginning of this effort to have a turnaround time from sample receipt to our laboratories to reporting of results to the subjects through third-party intermediaries of 24 hours. And we've been able to achieve this largely because of streamlining of each of these steps and the development of these kinds of software tools. Lastly, our biobank storage facility has enabled the storage of all identified positive samples as they are identified at -80 degrees. These currently number more than 500,000 to date. As you can well imagine, these samples have the important potential to be a resource for follow-on studies of epidemiology, demography, development of long COVID symptoms, and other manifestations. The product of all these high throughput efforts is summarized on this slide. The graph on the left depicts daily sample receipt and processing totals from late March 2020 to the present day. In all, more than 8.6 million samples have been tested in that time, as illustrated by the linear graph. And you can see pretty much what you would expect, the beginning of the pandemic here with very low sample receipts. We hit a maximum of somewhere between 70 and 80,000 samples per day in November of 2020 that started to taper off until the emergence of the Delta variant. And now, sadly, we're starting to pick up. Our New Jersey lab is currently processing an average of about 20 to 25,000 samples a day. On the right is a graph that depicts the time course of the CDC surveillance sequencing project. And again, mirroring what you've heard in both the popular and scientific press, we see the emergence of the Delta variant shown in purple here over time. Back in June, it was a relatively minor component compared to the Alpha variant, which was the initial major population, and has exploded to now represent in September 87% of all the viral variants of concern that we've sequenced to date. All the viral variants, indeed, not just those of concern. So this is an ongoing effort and is of critical importance in order to survey the population for new and emerging variants of concern that we will hopefully never see, of course, and those are being monitored across the country by us and 5 other centers. Our laboratory is currently equipped to sequence as many as 6,000 viral isolates per week and have identified 19,000 out of 42,000 cases classified as CDC variants of concern. So we only obviously sequence the positive samples and only those that meet certain criteria that I'd be happy to discuss separately if you're interested. Pardon me. Any pandemic response program cannot remain static but must constantly identify needs early as they emerge and innovate solutions to address them. In the tradition of our saliva test that you've seen before, IBX continues to develop tests and tools to streamline diagnosis and assess risk. We've developed workflows for pooling of saliva, shown in the first diagram on the top, and swabs as well in a separate workflow. And each of these shows the timeline from the design and validation of the assay through to product launch. A test to detect the presence of COVID antigen in saliva using antibody detection by agglutination PCR, or ADAP, is projected to go live this winter. This is being developed in collaboration with Enable Biosciences. As the flu season ramps up, it will be critical to diagnose and distinguish flu, COVID, or RSV infections in patients that present symptoms that often overlap or are in common. To this end, we're preparing to roll out a multiplex PCR test that combines the use of primers against all three. And finally, we've developed a COVID severity risk test that was launched in April of 2021. I'll have more to tell you about this later in the presentation. Through the example of our efforts to respond to this pandemic, you've seen an illustration of the mission and capabilities of IBX. In the next portion of this presentation, I'll provide an overview of our service offerings and expand on our mission. IBX serves a number of client bases in a variety of sectors. Clients in the biopharma field tend to advance their early stage drug discovery process, need to advance, their early stage drug discovery process and care deeply about regulatory and compliance issues, often needing a consultative, hands-on approach. IBX employs dedicated quality and scientific affairs teams to assist with these regulatory needs. Biopharma clients are also very technology driven and require a broad set of technology tools for which we provide evidence and validation, the validation assays being a key component in both research and clinical settings. The digital health field, on the other hand, involves getting their products to market quickly, often with the fastest and simplest hands-free solution. Flexibility and scale are important, as are simpler communication language. We share their concerns about capital expenditure costs and regulatory issues. We also see ourselves as a partner to assist them as they scale up in the entirety of their process. In academia, client needs tend to center on high integrity science, reproducible results, publishing novel findings, and are often constrained, unfortunately, by relatively smaller budgets. The ability to access the latest cutting edge technologies without CapEx costs is a major advantage for academic researchers, especially if the work can be performed faster and more efficiently than in their academic core labs, or even doing it in their own laboratories. This is, in fact, at the core of one of our missions, given our origins as an academic biorepository, where we recognize the vitality of academic research. Clients in the applied market sector care deeply about customization and often present with novel sample types, non-human, for example. Indeed, we've worked with many different species across the board in our various projects. In this case, they may care about fewer markets, but at scale and often operate as consortia. They often lack the requirement for the highest tech solutions that the other sectors rely on and are very sensitive to cost. Their needs tend to run on the high throughput side. So we have mechanisms in place for accommodating all of these different clients. Indeed, all of these clients, potential or existing, benefit from the availability of reliable partners such as IBX for outsourcing. This option presents companies with significant opportunities for streamlining projects and saving on their bottom line, which is at the end of the day, I think what everybody wants. To meet the challenges of a competitive market, providers such as IBX offer solutions that leverage their experience, infrastructure, and access to advanced technologies, allowing customers to focus on what they do best and offload the heavy lifting to us. Depending on need, and it's very variable from client to client, as one might imagine, we offer assistance in establishing scientific approach, scientific design, interpretation, power calculations, you name it. Indeed, IBX was founded by scientists for scientists. Licensure, we have the ability to provide medical grade services and are CAP biorepository accredited, as well as having CLIA license in a number of states for a variety of different tests. Flexibility, we take pride in meeting the individual needs of each project and designing custom workflows. We do not have cookie cutter SOPs. We work with clients to meet their individual needs. The ability to scale up rapidly, which was, I think, exemplified quite well by our pandemic response that I described to you, and technologies that push the envelope of the possible. But perhaps most important of all, we view ourselves as a partner and not a vendor in the client's project. Our services run the gamut from project management, including collection kit design and fulfillment, through sample processing, including high throughput nucleic acid extraction, quality control, and aliquoting on a large scale. Fractionation of blood to yield plasma and serum that are then analyzed by quantitative quality control assays that I would be happy to discuss if you're interested. Our cell culture and stem cell services include isolation and quality control of primary cells, including peripheral blood mononuclear cells from blood, fibroblasts from skin biopsies, and a number of other tissue types, as well as fluorescence activated cell sorting and ELISA characterization assays to examine cell subtypes within these various cell populations. Our advanced sample processing group performs reprogramming of induced pluripotent stem cell lines with all the associated quality control measures, as well as having a dedicated CRISPR editing team that will do editing in iPSC cell lines, and distributes iPSCs from the major collections we manage, such as those of NIMH and NINDS in the United States, including a GMP grade iPSC line. These are really valuable resources that are available. People often don't know about them, but they've been established by NIH as a resource for the world community. And I would be very happy to point you in the direction of how these can be ordered. And they're available equally to the commercial and academic sectors across the world, not just in the United States. The IBX stem cell group is also developing a set of neural differentiation workflows for offer, and details of these are available on request as well. Our genomics team services include all major genomics platforms, including DNA identity fingerprinting using a SNP trace 96 SNP assay developed in-house at RUCDR at the time in collaboration with Fluidigm. I should mention that this has become an industry standard assay for identity fingerprinting and tracing of sample identity through processing, and is done on all DNA extracted at IBX, all cell lines that are handled, produced, or cultured at IBX. It's a very valuable banking tool. Our genomics team also offers large-scale PCR, quantitative PCR, and digital PCR projects, as well as next-generation sequencing on all the major platforms, targeted whole exome and whole genome. Methylomics, using a variety of approaches, both array and next-gen sequencing, genotyping, and microarray studies. Assay design and molecular genetics diagnostic services, as you will see a little more of an illustration of later, are also offered. Our bioinformatics team works with clients in a highly customized and individualized way on experimental design consultation and a variety of other analytical, interpretive, and report generation services. Lastly, all of these resources rely critically on our CAP accredited biobanking facilities with regulated storage available at all manner of temperatures. I won't go into this slide in too much detail given the time, but I would be happy to make our slides available to anyone on request. This is a review of the clinical trial solutions that we have on offer. We have a dedicated project management team that works closely with our clients on all aspects of clinical trial management in collaboration with us. IBX offers, so let's start from the kind of the workflow, the chronology of a sample. So we offer all manner of support for sample collection, study design, and collection logistics. We have a number of GCP compliant kitting workflows. This just shows you the workflow of designing and fulfilling those kit requests. For the design, production, and distribution of kits for a variety of different samples, including pretty much anything that can be collected out of a human being: whole blood, urine, breast milk, saliva, stool, etc. So these are all assembled with all the components needed for collection and transport to our processing facilities, and are fully compliant with the transport regulations of each collection country involved. Once collected and received in our labs, samples can be processed along many different workflows, too many to mention in a talk such as this, depending on project specifications. Our sample processing labs, as I mentioned previously, isolate primary cells. One thing I did not mention is that we're also, we manage large cancer cell banks for Pfizer and Merck, and with all of the concomitant logistical issues associated with huge banks of several hundred cancer lines from different species with different media compositions. And this is one of the outsourcing capabilities I was referring to earlier. Included in our cell service capabilities are quantitative counting and viability analysis, critical in clinical trials with PBMC management. And cells are cryopreserved using computer-controlled rate freezers. Every vessel in the facility is barcoded as an individual unit, and are all stored in CAP-compliant monitored storage with GMP-compliant cold chain logistics for distribution. There will be a quiz on this later. No, I'm just kidding. This is a comprehensive list of the various omics capabilities that we have. Again, I won't go through this except to say that I'm happy to share lists of any of these services with you at the conclusion of the talk. At the core of our mission, and I think I've emphasized this point throughout, is the goal to always stay ahead of the curve in providing our customers with the most advanced and powerful tools to accomplish their goals. To this end, we're currently adopting and validating a number of exciting new technologies shown on this slide. The LabMan is a company in the UK, actually, that has developed a high throughput sample handling and liquid transfer system. It will take a saliva or a blood tube through the process, all the way from decapping to transfer to a processing plate at the rate of as many as 20,000 samples per day. We're in the final stages of validating this instrument in our laboratory, and we'll soon be putting it into operation. It's really a remarkable system to see working. Also, we're incorporating long-read sequencing platforms from ClearDX and PacBio into our menu of offerings. The ClearDX specializing in repeat sequences and difficult to sequence DNA regions. And we perform, as I said before, next-gen sequencing and Sanger sequencing using all the major platforms. We're currently installing a fully automated sample storage and retrieval system from Brooks, the BioStore, sample store, BioStore, that will increase the efficiency of our sample management process. We're very excited to bring in the 10x genomic technologies for single-cell DNA and RNA sequencing, as well as spatial gene expression, the Chromium and Visium platforms. In the case of Chromium, we're in the final stages of validation, and we'll be offering that soon. Visium is a remarkable system that will allow us to interrogate DNA or RNA sequence information in the context of the geography of a section of tissue, enabling oncologists, for example, to correlate genomic data with pathology, or neuroscientists, to give another example, to examine the patterns of expression across the developing brain. Two case studies are presented here for your consideration. As the pandemic continues to tax healthcare systems to their breaking point, we recognize a need to identify individuals at greater risk of developing severe symptoms. The IBX COVID-19 severity risk test was designed using a panel of 7 SNPs determined by correlative studies using UK Biobank data to be predictive of risk. This test is based on buccal swab collection and is now available. And these are some of the advantages to having a test of this manner, including allowing the subject to be proactive and take risk-based precautions and get vaccinated if they weren't already. We've developed another test to offer an accurate prediction of an individual's future risk of cognitive decline due to probable late onset Alzheimer's disease. This test is ordered in conjunction with the subject's physician, pardon me. The genotype of 112,000 SNPs is determined and imported into Cytox software to develop a polygenic risk score, additionally an apolipoprotein E status for the individual. And this is coming to market as we speak. The proof, they say, is in the pudding. This slide is a sampling of the clients who've entrusted us, some of the clients who have entrusted us with their vital projects and samples. We're very proud to be working with these leaders in the pharma, biotech, and academic sectors. And if you don't see your name on there, you're not forgotten. As I mentioned before, we're fully accredited as a CAP biorepository with CLIA licensing. We adhere to ISBR best practices, and we have a number of affiliations with global biobanking organizations. I'd like to take the last couple of minutes here. How am I doing on timing? Alicia, have you my
yes,
little timer?
Yeah, you're doing good, Mike. Yes.
Okay,
good.
terrific. Thank you. Okay, I'd like to take the last few minutes here before handing off to my colleague to tell you a little story, and it pertains to disaster recovery and another vital role that biobanks play. We had a natural disaster in this part of the United States in 2012. Hurricane Sandy hit the East Coast in a very terrible manner. And some of the pictures on this slide illustrate the after effects of that disaster. This is a picture of the Lower East Side of Manhattan after power had been lost. And as you can see from, if you're familiar with New York City, from about 36th Street on the east side to 29th on the west, all the way across the lower side of Manhattan. And that includes several important hospital and research facilities. Among them NYU Langone Medical Center, Bellevue, and the Veterans Administration Hospital. As a result of this, patient evacuations had to be made. There was flooding up to several levels in some cases in these institutions, and actually emergency generators had to be run at all hours of the day and night for several days in order to initiate the recovery. This was actually covered in an ISBR, International Society for Biological and Environmental Repositories newsletter at the time, what a dramatic effect this storm had on people's ability to conduct their research. So it was against this backdrop that we, at the time as RUCDR, in collaboration with our partners at Biostorage Technologies, actually rose to that occasion as well. And this was all started by our COO, Andy Brooks. And he's quoted here as having said at the time that the moment they started moving thousands of patients to other hospitals and medical centers, we knew there was going to be a problem with the biological and clinical samples. And we were obviously very concerned on a number of levels about people's research and all those precious irreplaceable samples. Now, the problem at the time was the logistics was a nightmare. Tunnels in and out of the city were down and flooded. Many bridges were being reviewed for safety. And it was actually a Herculean effort just to get a fleet of trucks, and actually managed by BioStorage at the time, that were equipped with temperature monitoring systems, dry ice and liquid nitrogen, into the city so that we could recover those samples. I was on one of these teams together with about 100 volunteers, and we worked for several days removing samples from the 17th floor in some cases without power. So we literally had to bring trays of dry ice up and down the stairs, accompanied by the New York City police on each trip. It was really quite a remarkable day. And we were able to recover the collections, including precious clinical samples, including some very highly hazardous samples, I might add. So that required expert training in handling and the logistics of dealing with there were actually some Ebola, hepatitis, and HIV samples in those collections. Loading them into the bunker carts, loading them into these reload fleet trucks that were monitored for temperature throughout their trip to either New Jersey or Indiana. So it was really a very remarkable effort that we're actually very proud of to this date. So biobanks aren't just banks. They really are partners in the global research community, and are there when people need us. And in fact, some of these institutions are actually now using IBX as their storage facilities, having recognized the need to outsource these kinds of capabilities to a CAP compliant facility that is able to store them in a safe manner. This is the global footprint and serves as an introduction to my colleague of IBX. We have facilities, as I mentioned, in New Jersey and Minnesota. We are very proud to have added to the IBX family, and I will not steal too much of his thunder by going through this slide, except to say that Ben Roylance will tell you about all of the wonderful accomplishments of Roylance Pharma. And I'd lastly like to leave you with an introduction to our executive team. A lot of these folks are pretty new because of the fact that IBX is only a year old, but are an amazing group of people with decades of experience in the industry. And we're very fortunate to have all of them on the team. And team is the theme. We are all IBX. And this is from our corporate brochure. It gives you a sense of who we are and how far we've come in a relatively short period of time. And with that, I'd like to transition over to Ben so that he can tell us about Roylance. And I thank you for your attention and look forward to your questions.
Brilliant. Thank you, Mike, for that. I'll just get my slides ready.
Oh, stop sharing.
Hopefully that's shared okay for everybody there.
Looks great. Mm-hmm.
Fantastic. Just like to say again a quick thanks to Mike for a really fantastic presentation there. Some really good things and just really interesting point to end on the disaster recovery. So our introduction, we're Roylance Pharma. We've recently been acquired by IBX, and I just wanted to pick up on that disaster recovery theme and run through it. Today I wanted to cover a few things, just look at the background of the company, just run through who Roylance Pharma are, what our facility looks like, the licenses that we hold, our disaster recovery solutions that we offer. A quick case study, there's one that's popped up in the BBC in the last week over here, back in the news, and then just some conclusion points around that. So who are Roylance Pharma? Who are we? So we're a fully integrated end-to-end storage solution for life science. We offer ICH stability storage, biobanking, outsourced validation, service contracts, and also disaster recovery services here throughout the UK. A quick overview of what we offer. So again, we're a combined service. We look as a fully integrated solution to our clients. So We've got the validation and service. So that's our technical engineers. They're out in the field looking after our clients, throughout the UK, but also looking after our old equipment. We've got our controlled storage and biobanking. So, we can store samples from stability, medical devices, cosmeceutical, veterinary, and so forth. And within our biobanking, we've got liquid nitrogen vapor phase, -80, -20, and all the disaster recovery services that sit underneath it. So I wanted to give a little timeline as well for Roylance of where we've come from and where we are. So 2016, we formed Roylance Scientific, and that was to offer the outsourced validation, service, and monitoring equipment for our clients. A year later, myself and my business partner, Marianne, formed Roylance Storage, and that really was what we've always aimed for, which is our outsourced storage facility, allowing us to take, pharmaceutical samples and biological samples. During 2018, we licensed the facility, so we put controlled drugs licenses in, schedules 1 to 4. We looked to build our quality systems with ISO 9001:2015, which is a UCAS accredited one, and started expanding our brand new storage facility. That theme continued into 2019, where we added GMO, so genetically modified organisms, for storage level 1 and 2. We had our RA inspection and added an IMP MIA license, and started then moving into more the biotech, so some of the -80 and -20 conditions. During 2020, we actually merged Roylance Scientific and Roylance Storage because we were working for the same clients, and we found that our clients, really are partners, and they were looking for a solution. They were looking for us just to take hold of the sample storage, and if they had a breakdown, come out and fix it. If it couldn't be fixed, transfer the samples into our facility. If they got stuck or they had a huge inquiry in, we could help them with that. So that's what really drove the merge to create Roylance Pharma. And again, the start of the pandemic, it pushed us into looking to get GMO level 3 so we could start to hold COVID samples, on site in our repository. And 2021, we've just been recently acquired by Infinity Biologics, which is fantastic. We've added our LN2 vapor phase, and we're currently working towards our HFEA license that we do hope we'll get by the end of this year. What do we look like? So quick insight here. This is inside our facility. So top left, you've got our document archive storage. So it's not just about storing samples. It's also important, to retain all your lab books and associated documentation and solid media. Our stability chambers are all custom built by ourselves. And Marianne has an ethos around color-coded baskets, so you can't get any mistakes there. Green, yellow, red, blue, denote different conditions and so forth. And again, in an emergency, when you haven't got time, it just allows you to work visually. And again, it's all these themes that we're running through. All our equipment we use the same equipment over and over again. So you'll notice our monitoring systems, our control panels, our chillers, and this is all around disaster recovery. We can have multiple supplies in stock, and should the worst happen in our facility, we've got spares available immediately. And the top left, a couple of our staff there, and it's just everything we do is all a minimum of a double check. So any samples that are coming in, samples that are going out, if we're collecting on a disaster recovery point, we'll double check. So it's always, criticality. We're looking after the sample at every single stage. Our regulations. So again, just to roll into what do we have? So MHRA license, MIA, IMP, GMO. So we've got BSL levels 1, 2, and 3. It's just to note that 3 at the moment is just for storage only, and it was really just in reaction to the COVID pandemic and to be able to store some of those samples. We've got controlled drugs license in place, so schedule 1 to 4. Our ISOs we touched, and we're also now a counter-terrorism approved facility. We have them in sort of every 4 to 6 weeks now to come and inspect and audit our facility. And for us, we're always striving for excellence. So it's always looking towards the next, regulation, the next license that we can achieve. And the team are working towards our HFEA, which we expect by the end of this year. And during the next year, we're actually working to get our WDA license. So the next part is I just wanted to maybe take a few slides to into disaster recovery and just talk about risk management. And just before you start with disaster recovery, it's really all about your business continuity plan. You need to make sure you've got a business continuity plan in place. In the event of an emergency, you want them to have a clear, concise plan. Everyone's going to be running around. Everyone's going to be worried. There might be fire engines on place. There might be floods. You need to know what you're doing. You need a document available. And consideration to a business continuity plan really needs to look at the people. Who are the key contacts? Are these key contacts regularly reviewed? Do you make sure you keep all the information up to date, emails, phone numbers, if staff move on? the premises, again, recovery operations, having a look at, where are your samples kept? Where's your IT kept? Where's your documentation? Is it accessible? How do you get to it in the event of, loss of power, fire, flood? The processes. So again, as a business, you want to look at how are you going to return the organization back to at least minimal service levels, even if you have to rely on some of your outsourced partners to help you. And announcements, you might be multi-sited. So, announcing to staff that things are in control, how things are going. And again, if you have to talk to your clients, how do you want that presented? These are things that you want to think about, before the event. And then the last one is suppliers, and that's sort of where your disaster recovery supplier comes in. That you've got your key sort of supply chain suppliers. So again, evaluate them, risk them, and then look to mitigate any risk in that supply chain. So moving on from a business continuity plan to disaster recovery, again, it's this early stage where you want to identify all the potential risks of your storage program, identify their impacts. We'd always say and recommend you do this via risk assessment. And then again, just highlighting that the risk or loss should not just include your samples. There's other associated documentation, electronic means and elements, hard drives, lab books that may sit around there. These samples may have been in storage for 10, 15, 20 years, so there could be a lot of ancillary documentation to go around them. So, things to think about and consider: damage or complete loss of samples, damage or complete loss of electronic data. These can, create their own SOPs and work instructions for each element. Access to the samples. If the samples, as Mike alluded to, are on the 17th floor and the power's out and there's no lifts, how are you going to get those samples down? Are they, as I say, temperature sensitive? Do you need to bring dry ice? And there's all these elements that you just need to consider, in the plan and just work the way through. And then just a clear route. Where are you going to bring the samples to? Is it down to a car park, to a road? Like you have a fire point, you really need a point where you're going to meet your supplier and bring your samples to, so that they can be collected, packed, and taken away for long-term storage. So how do you choose that? Where do you start with choosing the right disaster recovery provider? The way that I think you should do this is through a physical audit. So identify who you want to work with and go to their site, have a look at their quality systems, their staffing capacities. Look at the employee training records and programs. Look at their CAPAs and deviations. What corrective and preventive actions have gone in there, and document control. With a real focus, looking at all the storage related elements. And then when you're on site, have a look at their chambers, the capacities. How much storage do you require at LN2, -80, -20? What security is there, and alarms and backup systems? Do they have a UPS in there? Do they have dual power supplies? What's their plan? And what does their disaster recovery plan look like? So you're asking for one, but your supplier should also have one. So what happens if they, we have fire, flood? What happens to your samples at that point? Should they be on place? And that operator and provider should be able to give you all that. Another bits that you might touch on is, the independent monitoring systems, the qualifications, so looking at the validation, the IQ, OQ, and also having a look through the relevant guidelines and staff knowledge. You really have to get comfortable. This client relationship is a lot of trust. In your hour of need, your disaster recovery partner is going to be that person who's going to ensure, your critical sample survival. So again, different options that providers can give you. You can have retention only. So do you just want to reserve some space in a freezer or a fridge? Do you want to look at logistics? Do you need your samples collecting, and those samples bringing from the different sites up to your provider? Do you want to look at it in a different way and maybe split stock? So rather than having all your eggs in one basket, actually you're going to put half your samples, and split your master cell bank or something, and push those samples off site. So you've got a continuation. And response times, how quickly, do they need to be down in 4 hours, 12 hours, 24 hours? And again, the way that we would work with our clients, we look to be a service provider and extension. So we'll work with your validation. We'll look at power failure simulation tests. So how long you've got for your -80 or liquid nitrogen tank is hitting criticality points, and those samples, are potentially going to start to get damaged. So it's working all that in and then coming up with a disaster recovery plan. So I was watching the news about a week ago, and actually this came on, and I thought it was a great example. Back here in the UK in 2017, the Christie's Hospital actually caught fire. And it was recently brought back into the press on the 27th of September this year, when they identified it was actually caused by a welding issue. So they were welding the roof, Some of the well dropped down the side. They checked the sides, everything was clear. They left, and about an hour later, it had been smouldering and it set fire to the reef. At this point, we don't know whether the Christies or not had a disaster recovery plan in place, but having a look at everything, some of the quotes in the press was that the Institute was very grateful and happy that the fire service helped salvage its tissues samples, which are stored at -80 and -200, but it's -196. But my question is, they brought all these to the car park and salvaged them, but what happened next? The -80 could have gone to another building, but the liquid nitrogen, that's a really specialist, storage, and how quickly was that response? So I think it was quite pertinent that, in the event of emergency, you need a very clear, concise plan, to follow. You need to be able to get hold of your provider, make sure that they cover, all the conditions that you require. And, it's not only the maintenance of your equipment, but it's also looking further than that and just ensuring with the suppliers and hot work permits and all the other parts that literally it can go wrong anywhere. And we look at it as an insurance policy. So my take-home points really just around disaster recovery are, keeping your documentation, all the contact details up to date. So regularly review your documentation. Ensure you've got enough capacity. A lot of our clients are growing really quickly and they're adding freezers and freezers and freezers. What you don't want to do is hit that point and suddenly realize we can only store 5 freezers and you require 10. Responsiveness, ensure that you've done all your background work and looked at, is your supplier that you're working with responsive enough? Are they going to react within 4 hours, 12 hours, 24 hours? Consider your logistics. So it is one thing reserving the space, but again, just consider how you're going to get those samples, to that provider that could be 4, 5, 6 hours away. Validate your plan. So we always sort of push on, run through it, test it, make sure it works, rework it, change elements if they don't. Ongoing training with the staff. So train your staff, in your disaster recovery plan. Make sure it's all in one document, electronically or physically. It's a grab and go. It's an emergency. You just want to be able to take that document, find the numbers you need, and enact that plan. So my conclusion personally is that, a successful disaster recovery plan comes from successful collaboration, between client and supplier. The information in the disaster recovery plan, the resulting documentation is paramount to being a success or really a failure. Evaluate your disaster recovery plan partners closely. Look at what samples should be stored, all the ancillary documentation. And, most importantly, again, I feel is get it all in one place. If you're in an emergency and people are rushed and worried, you just want to be able to pick that plan up and go. Stay in regular contact with your provider. So, monthly, quarterly meetings, update samples, documentation, protocols, and again, staffing changes on there. So everything's up to date, and once you ensure you've got that right plan in place, that right partner, that has the capacity, responsiveness, and technical considerations that you require, it is possible to mitigate your sample operations, minimize disruption. And to be honest, all we're trying to do is ensure the integrity of your replaceable samples. So Mike touched on, obviously, the New Jersey site, and he very kindly sent me a slide. So I just wanted to sort of touch on, our capabilities now, both sides of the Atlantic. We're hopeful to get our LN2 repository to this size within the next sort of 6 to 12 months. But it does mean that we offer disaster recovery services now globally. And again, just to jump on Mike's slide, which I did steal a copy of, is Minnesota, New Jersey, and Glasgow are all IBX sites. We have partner sites as well now in Holland, China, and Australia. So we can look after you on that service level as a global presence. Thank you for your time. Hopefully that was useful, and I just about, I think, kept to the 15 minutes.
Excellent. Thank you very much indeed, Mike and Ben. Both really interesting, excellent presentations. Mike, I particularly enjoyed the case studies you presented there, and Ben, really important considerations, which I don't want to speak on behalf of anyone, but maybe sometimes things aren't assessed up front. So some really important considerations there. Does anyone in the audience wish to unmute to ask any questions? We are up to time, but I understand our speakers aren't in any hurry. So
I have a question or two, if I may.
Of course. Sorry, I couldn't see who that is. So yes, please.
I'm sorry, it's Tim Hines.
Please go ahead. Thank you, Tim.
Both presentations, I enjoyed both. Thank you so much. Benedict, maybe I'll start with you. How often do you folks back up your system or test your backup system, if I may?
So we've got different elements on different systems. So we've sort of diesel backup generators and so forth, which we might do on a monthly basis. And then you have like your daily checks, your weekly checks, your monthly checks, and then we've got sort of the annual servicing. So there's lots of elements, Tim, that go into it, depending if you're talking about a fridge or a freezer or a stability chamber. A lot of our equipment that we have on site as well has all got dual redundancy in. So our 5 degrees have an A and a B system, and they can run on either. And the same in all the freezers is an A and a B. So if you do get a failure, there'll be an automatic fallover.
Very good. Okay, thank you. Thank you. If I may, I have one other short question for Dr. Sheldon. Very nice presentation as well. Thank you so much.
Thank you, Tim.
You mentioned over 500,000 samples in storage, COVID positive samples. Now, I know they may be anonymized, but if clients wanted those samples for upcoming clinical trials, whatever, is there any issue with informed consent or anything like that? That may be a challenge.
Right. No, that is a very good question. All these samples are collected under IRB approved protocols that include a patient consent. So they've signed that over, and it's been explained to them what all the implications are of their samples being used in research. As you say, they're anonymized. So we, as a biobank, we don't retain any of the individual information. We have no HIPAA sensitive information on any sample. They're all coded as far as we're concerned with just a subject code. The key to that code is kept by the third-party providers like Vault Health. So any kind of clinical follow-on studies would involve a collaboration with them, and they're approved to release that clinical information as it's needed to interpret the data. But yeah, that's a very good question. Those are free and clear.
Very good. Thank you. Thank you.
Sure.
Thank you very much for your questions, Tim. And thank you once again, Mike and Ben, for presenting. Thank you very much for the attendees who joined. I will be sending a link around the recording. If there's any specific requests for the presentation slides separately alongside that, please do let me know, and I'd be very happy to send those out on behalf of Mike and Ben. I'm going to stop the recording shortly, but just to highlight that if anybody does want to remain online to speak directly with Mike and Ben, then I believe we still have some time. And I think I've just seen a delegate join at the end of the presentation. So perhaps if you do want to stay online to ask any questions, then absolutely no problem. So I'll just say thank you very much to everyone again for joining, and I hope you found it a valuable and useful session. Thank you.